Much. Good morning, everyone. As Michael said, my name is Brenna Levine, and I'm a graduate student at the University of Arkansas, where I specialize in molecular ecology and conservation. And paramount to conservation decisions is an understanding of the relationships among populations and the ecology that underlies those relationships. And today, I will be providing an example of that in the eastern collared lizard in Arkansas. The eastern collared lizard is the largest and, for my money, the most beloved species of lizard in Arkansas. It was once historically common in Arkansas, spread across a patchwork of rocky glade habitat in the state. This is a heliothermic species that relies on those rocky glades for thermoregulation. However, the reliance on this habitat has now resulted in this species becoming one of conservation concern. The way that rocky glade habitat should look is on the left here, where you have a whole bunch of barren rock, ideal for thermoregulation. However, fire suppression over the years has resulted in the growth of forest where rocky glade habitat once was. And so, where we once had high genetic connectivity among these populations, with lizards being able to move from rocky glade to rocky glade fairly unimpeded, we now have a situation where our populations have become habitat islands. So habitat islands behave very much like actual geographical islands in the ocean, for instance, where we have limited gene flow among those islands and are expected to have elevated levels of genetic drift or random loss of alleles over time due to a lack of new alleles coming into the population. And over time, habitat islands then become subject to local extinction. And in fact, we'll see at the end of our talk here that this has already happened in one of our populations during the course of our study. Conservation of the collared lizard in Arkansas is currently relying on two main methods, and those are habitat restoration, or restoring our rocky glades via controlled burns, or via translocations, where we are moving collared lizards from healthy populations to newly restored habitat, or moving them to populations that are genetically depauperate to try and increase their genetic diversity. Our research group has recently done some work on the historical genetic connectivity of the collared lizard in Arkansas, where we've seen through mitochondrial DNA analyses that historically, and I'm talking pre-European settlement here, historically, all of our different populations really behaved as one evolutionarily significant unit because there was a lot of rocky glade habitat for lizards to move from habitat to habitat. And there was low genetic divergence among these populations. And we saw this even though we were looking at three main regions as depicted with the different colors on this map. However, to really inform translocation decisions and to prioritize areas for habitat restoration, we need to evaluate contemporary connectivity or more recent genetic differences among populations. And so that's what our objective is here today. I'm going to be describing how we evaluated contemporary connectivity of the eastern collared lizard in Arkansas. And our goal was to answer three main questions. First, how genetically diverse are populations of collared lizard? Genetic diversity is the raw material for evolutionary change and the ability of a population to adapt in response to evolutionary change. And so this is a very important metric for conservation. Secondly, we wanted to evaluate how genetically distinct populations of collared lizard in Arkansas are. Do we have one management unit or a bunch of distinct management units that need to be considered separately? And finally, if we do have genetic distinctions among populations, is this purely due to the distance between those populations, as we would expect in sort of a stepping stone island model where lizards can move from habitat to habitat relatively easily, or is this more due to something like genetic drift? Over 5 years, we sampled DNA from 282 adult collared lizards. My colleague, Casey Brewster, who's presenting tomorrow, has collected these collared lizard samples from 15 populations in Arkansas. And these are the same populations that we evaluated during our mitochondrial DNA study, with the exception of one population that was represented by a single lizard for this project. Overall, that resulted in a mean number of samples per population of about 18.5 lizards. Don't ask me what a half lizard looks like. I think it's probably terrifying. We genotyped each individual at 10 polymorphic microsatellite loci. For anyone unfamiliar with microsatellite loci, they are co-dominant genetic markers that are ideal for studying gene flow and genetic drift. And then we evaluated genetic diversity and connectivity measures. Specifically for genetic diversity, we looked at the number of alleles per population, or the mean number of alleles at each locus in each population, also known as allelic richness, and our expected heterozygosity, or the proportion of individuals in each population that have 2 alleles at each locus rather than are homozygous. To evaluate connectivity, we performed a genetic Bayesian assignment test with program structure. And we also evaluated pairwise FST, or genetic differences between populations. To give you a quick idea, a pairwise FST between 2 populations of 0 would mean that they are the same population. Genetic divergence of 1 would be that they are completely isolated from one another. And this was visualized with a PCOA. And then finally, we conducted Mantell tests in order to evaluate whether or not we do have isolation by distance among populations. With regards to how genetically diverse our populations are, overall, we had a relatively low allelic richness among populations with a moderate expected heterozygosity. But this is really most informative if we look at where our lowest and highest values are on this map here. Our lowest expected, or our lowest mean number of alleles was found in a very isolated area. So in our population here in Logan County, we are surrounded by dense forest and very limited amounts of collared lizard habitat, if any. Our ground truther, Casey Brewster, who's done all the ecological work for this project, hasn't seen any collared lizard populations nearby. And so it's not surprising that over time, we would end up with a loss of alleles in this population due to genetic drift. Our highest allelic richness, on the other hand, was found in an area where we have a more dense population of collared lizards, a more dense cluster of populations. Similarly, our lowest values of expected heterozygosity were also found in an isolated area. This isolated area is in Carroll County, near Lake Leatherwood, and once again, we don't have suitable habitat for collared lizards around these populations. I want to make a quick note that we also had extremely low levels of heterozygosity in Logan County. I just chose the one that was the lowest for this example. And to reiterate, our highest heterozygosity, once again, a measure of genetic diversity, was found in this area where we have more collared lizard populations. We absolutely have genetic structure among populations in Arkansas, which means that we have distinct genetic units that are evolving separately at this point. And so we can see that we have 8 distinct genetic units, and I'll show where these are on this map here. Those populations which are most isolated, those in Logan County, Carroll County, Newton County, Marion County, all represented distinct genetic clusters. Differently, if we zoom in on our high amount of populations that are along Sylamore Creek in Arkansas, we have much more connectivity. We still see genetic differences. I really want to point out this population here in Stone County. It's very close to all the other populations in along the Sylamore Creek, but it is separated by some dense forest. Even a small patch of that forest is preventing lizards from moving to the next population. With regards to our FST analyses, how much pairwise differentiation we have among populations, the majority of all of our pairwise tests among different populations were statistically significant. And we did use a von Ferroni adjusted alpha for this to adjust for multiple comparisons. And you can see that at a broad scale, we actually end up with clumping on our PCOA that roughly corresponds with the location of these different populations in Arkansas. If we look at a zoomed in area just along that Sylamore Creek, we still see that we have statistically significant differentiation among those populations. So the majority of those pairwise tests were still statistically significant, even at that local scale. Now, whether or not this was due to the distance between populations, we found no support for that hypothesis. So a Mantel test tests the null hypothesis that there is no relationship between geographic distance between populations and genetic distance between populations. And we did fail to reject that null hypothesis. And so it seems that there's not suitable habitat between populations where there may be pockets of pollard lizards that are allowing us to see that relationship. And this same pattern, or lack of pattern, I should say, was also found at a local scale. So what these results point to is exactly what we would expect if we were studying islands in island biogeography theory. We see that we have low levels of gene flow even among nearby populations that are separated by inhospitable habitat or bad thermal environments for collared lizards. And over time, the loss of genetic diversity in these populations due to genetic drift has caused them to drift away from one another. In fact, one of our populations has already become extinct during the course of our study. This specific population in Marion County, which we had a very low sample size of, is now gone. We haven't been able to find it since. And so our study really points to the importance of evaluating genetic connectivity and structure in order to inform conservation plans. So as an example of this, we have already completed one translocation prior to the start of this study. And without evaluating that genetic connectivity, we saw, looking at it now, that we actually moved a lizard from a population that was extremely different from the population that it was moved to. And that was found with both our Bayesian assignment test and with our pairwise test. And while this could be a good thing by introducing new alleles for evolution into the population, it also runs the risk of us introducing problems such as genetic swamping and outbreeding depression. And with that, we have many collaborators. I encourage all of you to go to Casey Brewster's talk tomorrow, where he'll focus on the ecological aspects of this project. Thank you so much. Yes, Rocky.
So I have a quick question about what are some of the life history characteristics that lead to this isolation on such a small geographic scale?
Right, so a lot of this Casey's going to be talking about during his talk tomorrow, but I'll give a brief. Most of it has to do with the specificity of this lizard to the habitat. Yeah, it requires that really hot thermal environment.
And I have a question.
Yeah.
So really isolated patches, how ancient is that? Is that anthropogenic, purely?
Yeah, so when we were looking at our mitochondrial DNA, that original study that I alluded to, mitochondrial DNA shows us a much longer scale, way before Europeans arrived in the United States. Looking at microsatellite DNA, we're looking more in like the last couple hundred of years.
Okay,
Yeah.
so it looks like primordially there was not a lot of structure there, but there's
Right.
some structure there.
Absolutely, bingo.
Okay, so you can, and that was a great talk, by the way.
Oh, thank you so much.
Yes, now I need to go to Arkansas.
Come visit.
My gosh, those lizards are so cool looking. Oh my gosh! But so really, you're not violating historical population structure by your translocation.
Right.
It's all about the genes. So,
Right, absolutely.
That's great. We have time for maybe one more question. You nailed it.
Excellent. Thank you so much.
Thank you.